We're going to be starting on chapter 17, where we are going to be discussing the blood. Most cells of a multicellular organism can't move around to obtain oxygen and nutrients or to eliminate carbon dioxide and other wastes. So instead, these needs are met by two fluids, blood and interstitial fluid. Now, blood is a liquid connective tissue that consists of cells surrounded by a liquid extracellular matrix. Now, the extracellular matrix is called blood plasma, and it suspends various cells and cell fragments. Now, the other fluid, interstitial fluid, is the fluid that bathes body cells, and it's constantly being renewed by blood. So interstitial fluid is completely surrounding all cells at all times. Now here on the left in this picture, we have a tube of centrifuged blood. So if a sample of blood is centrifuged, so it's going to be spun down, and it's in this small glass tube, the cells, which are more dense, will sink to the bottom of the tube. So you can see red blood cells here, and the plasma, which is less dense, forms a layer on top. Now, blood is normally about 45% formed elements and 55% plasma. Now, these pale, colorless white blood cells, and even platelets here, these occupy less than 1% of the formed elements. Formed elements just means cells and cell fragments. Now, because the white blood cells and platelets are less dense than red blood cells, but more dense than blood plasma, they form this thin buffy coat. This is a buffy coat layer between the packed red blood cells and the plasma in the centrifuged blood. Now, whole blood has two components. Plasma, which is a watery liquid extracellular matrix that contains dissolved substances, and formed elements. So these are going to be the two elements of blood. Plasma and formed elements. Now, formed elements, these are going to be the cells and cell fragments. Now, the formed elements, or the cellular elements, you can call it even, this includes red blood cells, white blood cells, and platelets. And the plasma consists of water, proteins, and other solutes too. Now, when formed elements are removed from blood, there's this straw-colored liquid or pale-colored liquid called the blood plasma. And blood plasma is about 91%, 91.5% water, and about 8.5% solutes, most of which are proteins. But then there's a little bit of other solutes, things like electrolytes, nutrients, gases, waste product. These are just other stuff that's dissolved in the plasma. Now, some of the proteins that we find in blood plasma are also found elsewhere in the body, but those that are confined to blood, these are called plasma proteins. Now, hepatocytes, which are the liver cells, these synthesize most of the plasma proteins. These are going to include albumins, about 54% of plasma proteins. Globulins are about 38%, fibrinogen about 7%. So these are all made in the liver by the hepatocytes. The formed elements of blood include these three principal components: red blood cells, white blood cells, and platelets. Now, red blood cells, these are also known as erythrocytes, just labeled here as erythrocyte. These transport oxygen from the lungs to body cells, and they also deliver carbon dioxide the opposite way, delivering carbon dioxide from body cells to the lungs to be expelled. White blood cells, also known as leukocytes, these protect the body from invading pathogens and other foreign substances. Then we have platelets. See them here, a little bit bigger here. This is the final type of formed element. These are fragments of cells that do not have a nucleus. Now, among other actions, platelets release chemicals that promote blood clotting when blood vessels are damaged. So this is the huge main role of platelets, is to act in blood clotting. Now, blood has three general functions. First one is transportation. So blood transports oxygen from the lungs to the cells of the body, and it also transports carbon dioxide from body cells back to the lungs for exhalation. It also carries nutrients from the gastrointestinal tract to body cells, and hormones from endocrine glands to other body cells. Now, blood also transports heat and waste products to various organs for elimination from the body. So a lot of transportation happening within the blood. Regulation is the next general function of blood, because circulating blood helps to maintain homeostasis of all body fluids. So regulation is our second main function. So blood helps to regulate pH through the use of buffers. Recall that buffers, we briefly talked about buffers before, but buffers are chemicals that convert strong acids and bases to weak ones. So that's one of the other things that blood contains within it. It contains buffers that help to regulate the pH of blood and body fluids. Now, blood also helps to adjust body temperature through heat absorbing and cooling properties of the water in blood plasma, as well as its variable rate of flow through the skin, where excess heat can be lost from the blood to the environment. In addition, blood osmotic pressure influences the water content of cells, now mainly through interactions of dissolved ions and proteins. Next function is protection. So that's going to be, here's our last function here. Now, blood can clot. Clotting means it becomes like a gel-like substance. And this protects against excessive loss of blood from the cardiovascular system after an injury. Now, in addition, the white blood cells within blood protect against disease by carrying out phagocytosis. Now, there are several types of blood proteins, including antibodies and interferons, complement as well, complement proteins. These all help to protect against disease in a variety of ways. And we will talk about antibodies and interferons and complement when we talk about the immune system. So this table here gives the substances within blood plasma. So we have water, the plasma proteins, and other solutes. Gives a nice description of each of these as well as their function. So again, a nice table here for studying. Now, although some lymphocytes have a lifetime measured in years, most formed elements of blood last for only hours or days, sometimes weeks, and these formed elements of blood have to be replaced continually. Lymphocytes, these are a type of white blood cell. So single type of white blood cell can sometimes last years, but most types of blood cells only last for hours or days. Now, negative feedback systems will regulate the total number of red blood cells and platelets in circulation, and so their numbers really remain quite steady. The abundance of the different types of white blood cells, though, this is going to vary in response to challenges by invading pathogens and other foreign substances. So because the white blood cells are a major component of your immune system, depending on what your immune system is fighting at the time, the numbers of white blood cells will vary. Now, the process by which the formed elements of blood develop is called hemopoiesis. Now, red bone marrow, this is a very highly vascularized connective tissue located in the microscopic spaces of spongy, or the microscopic spaces between the trabeculae of spongy bone. And some red bone marrow cells are called pluripotent stem cells. So these are found within red bone marrow. Remember that red bone marrow within the trabeculae of spongy bone. And these pluripotent stem cells have the capacity to develop into several different types of cells. So here we can see these pluripotent stem cells at the top of this diagram here. Now, in order to form blood cells, these pluripotent stem cells in red bone marrow produce two types of further cells. And these are also going to be stem cells. And these two stem cells have the capacity to develop into several other types of cells. So these two other stem cells that the pluripotent stem cell will develop into are either myeloid stem cells or lymphoid stem cells. Now, myeloid stem cells begin their development in red bone marrow, and they give rise to red blood cells, platelets, mast cells, eosinophils, basophils, neutrophils, and macrophages. Now, lymphoid stem cells, which give rise to lymphocytes, these begin their development in red bone marrow, but they complete their development in lymphatic tissues. So these ones are going to complete development in lymphoid tissues. Specific tissues and organs of the lymphatic system, which we'll discuss it in a little bit more detail when we go over the lymphatic system. Now, during hemopoiesis, some of the myeloid stem cells will differentiate into progenitor cells. So these are labeled as the green. So these are kind of lettered cells we can see here. So these are going to be these precursor cells, progenitor cells rather, I guess. And in the next generation, the cells are called precursor cells. These are also known as blasts. So proerythroblast, these are going to be the precursors to erythrocytes, which are red blood cells. Megakaryoblast are the precursor cells to megakaryocytes, and so on and so forth. Okay, red blood cells, remember these are also called erythrocytes. These contain the oxygen-carrying protein hemoglobin. And this is the pigment that gives whole blood its red color. So this is an important little protein here. Now, each hemoglobin has an iron ion, which allows the binding of 4 oxygen molecules. Now, red blood cells are biconcave discs. They also have no nucleus, and mature red blood cells really have a very simple structure. They don't really have any other organelles either. Their plasma membrane is very strong, but also very flexible, which allows them to deform without rupturing as they squeeze through really narrow blood capillaries. Because blood capillaries are actually smaller in diameter than red blood cells. So red blood cells have to kind of fold in on themselves to go through really small capillaries. So they're flexible enough to do that. Now, as we will see later, there are certain glycoproteins that are kind of sticking out in the plasma membrane of red blood cells that are called antigens that account for the various blood groups such as the ABO blood group and the RH blood group. Now, you can also see here that red blood cells lack a nucleus and other organelles, and so they can't reproduce, and they really don't carry out really many extensive metabolic activities. Now, within the cytosol of red blood cells, we have hemoglobin molecules. Now, these important molecules are synthesized before the loss of the nucleus during red blood cell production, and they even constitute about 1/3 of the cell's weight. Now, each red blood cell contains about 280 million hemoglobin molecules. Each cell contains about 280 million of these hemoglobin molecules. Now, each hemoglobin molecule consists of a protein called globin. Globin is composed of 4 polypeptide chains and also a ring-like non-protein pigment called a heme. And there's going to be 1 heme bound to each of the 4 chains within a globin. And at the center of each heme is an iron ion. So we can see this on the right here. So I have this iron ion right in the middle of the heme ring. And the iron ion can combine reversibly with 1 oxygen molecule. So this combined with just 1 oxygen molecule. So this allows each hemoglobin molecule to bind 4 oxygen molecules, because you get 1 bound here, 1 bound here, 1 here, and 1 here. In addition to its key role in transporting oxygen and carbon dioxide, hemoglobin also plays a really important role in the regulation of blood flow and blood pressure. The gaseous hormone nitric oxide, produced by the endothelial cells that line blood vessels, can bind to hemoglobin. Now, under some circumstances, hemoglobin releases nitric oxide, which then goes on to cause vasodilation. Recall this is an increase in the blood vessel diameter that occurs when the smooth muscle in the blood vessel wall relaxes. So if we're looking at a cross section of a blood vessel here, vasodilation would be an increase in the diameter of the blood vessel. Now, vasodilation really improves blood flow, and it also really enhances oxygen delivery to cells near the site of the nitric oxide release. Now, red blood cells also contain the enzyme carbonic anhydrase. Often this is just abbreviated CA. Carbonic anhydrase catalyzes the conversion of carbon dioxide and water to carbonic acid, which in turn will dissociate into hydrogen ions and bicarbonate ion, which is HCO3 negative. So this now bicarbonate ion allows about 70% of carbon dioxide to be transported in the blood and transport it from tissue cells to the lungs. Now, bicarbonate ion also serves as a really important buffer in extracellular fluid. Remember that buffers, like bicarbonate ion, are going to convert strong acids and bases into weak acids and bases. So that a strong acid or base that is found in the blood won't be able to change the pH of the blood significantly. It'll be buffered so that it does not change the pH a lot. Because if you change the pH of your blood, that could be deadly, really. So buffers are very important. Red blood cells really only live about 120 days. And this is because of the wear and tear that their plasma membranes undergo as they have to squeeze through these blood capillaries. Now, without a nucleus or organelles, red blood cells can't synthesize any new components to replace the damaged ones. So with age, the plasma membrane becomes more fragile with the age of the red blood cells, not the organism's age, but with the age of the red blood cells, the plasma membrane becomes more fragile, and as these red blood cells get older, up to like 120 days, the cells are more likely to burst. Now, ruptured red blood cells are removed from circulation and destroyed by phagocytes in the spleen and the liver. The breakdowns are reused in a lot of different metabolic processes, including the formation of new red blood cells. So really, destroyed, old and dead red blood cells are kind of recycled to produce new red blood cells. So when your body needs new red blood cells, this is when erythropoiesis occurs. Erythropoiesis is the process of producing new red blood cells. Now this begins in the red bone marrow. Reticulocytes, which is kind of the official name for immature red blood cells, will enter into the circulation as immature red blood cells, and they mature within circulation in a couple days. Now, when there's low oxygen concentration or hypoxia, the kidneys will detect this and will release the hormone erythropoietin, which will stimulate the differentiation of those hematopoietic stem cells into erythrocytes. So this is essentially your body's way of saying we're really low on red blood cells, we're not getting enough oxygen to tissues, so let's increase the number of red blood cells so that we can increase the transport of oxygen to tissues. So here we have that feedback loop. So we have a decrease in oxygen delivery to the kidneys, and really it's to like all of your tissues. You're having decreased oxygen delivery to all your tissues, but it's just going to be the kidneys that kind of detect this. And so the kidneys kind of think, well, if we're detecting low oxygen, there must be low oxygen everywhere, so we need to do something about it. So the kidneys detect these low oxygen levels and they synthesize and secrete erythropoietin into the blood. Now, erythropoietin will cause those pro-erythroblasts that we talked about earlier in the red bone marrow. It causes them to mature really quickly into reticulocytes. Now, even though the pro-erythrocytes will mature into reticulocytes, reticulocytes are still immature. So they're more differentiated. So now they've kind of chosen we are going to be red blood cells, but we still need to mature a little bit until we become full erythrocytes, mature erythrocytes. So as these proerythroblasts turn into reticulocytes, they go into the circulation. And then we have a lot more reticulocytes enter the blood. And like I said, these red blood cells just take a day or two to fully mature until they can fully transport oxygen. And the response is that there's increased oxygen delivery to tissues, and then we return back to homeostasis. And then when the kidney cells no longer detect that decreased oxygen delivery, then they're going to stop secreting erythropoietin. Now, unlike red blood cells, white blood cells, or leukocytes, have nuclei and they have a full complement of other organelles, but they don't contain hemoglobin. Now, white blood cells are classified a couple different ways. So they can be classified as either granular or agranular. Now, this depends on whether they contain these very conspicuous chemical-filled cytoplasmic granules or vesicles that are really made visible by staining when they're viewed through a light microscope. So granular leukocytes have these very visible vesicles, whereas agranular leukocytes don't have these granules. Now, granular leukocytes, these are going to include neutrophils, eosinophils, and basophils. And agranular leukocytes include lymphocytes and monocytes. So we'll see pictures of them and you can kind of see these granular filled vesicles. Now these white blood cells have been stained with an acidic and basic stain. Now if they are attracted to the basic stain, they will stain blue, and if they're attracted to the acidic stain, they will stain red. So after staining, each of the three types of granular leukocytes display these really conspicuous or visible granules with distinctive coloration that we can really recognize under a microscope. So first off here we have the neutrophil. The granules of the neutrophil are going to be smaller than those of other granular leukocytes. They're also going to be very evenly distributed. And they're going to be like a pale lilac color. So we can see these little granules all around here, all throughout the cytoplasm, very evenly distributed, very light colored. Now, because the granules don't really strongly attract either the acidic or basic stain, these white blood cells are neutrophilic, which means they are neutral loving. That's where they get their name from here, neutrophil. You'll also see that the nucleus has 2 to 5 lobes. This one here has 3, and these lobes are connected to each other with very thin strands of nuclear material. So we can kind of see these connections here and maybe here as well. So these connections to 3 lobes of the nucleus. Next, we have the eosinophil in the middle. Now these have very large, uniformly sized granules, and these granules are eosin loving. So eosin is the red-orange acidic dye. So eosin is acidic, and so it's going to stain red. So you can see these are a lot more reddish granules than either the neutrophil or the basophil. So these granules generally don't cover or obscure the nucleus, which often has 2 lobes. And they're usually connected by a strand, could be thin or a thick strand of nuclear material. We can kind of see this thick strand of nuclear material right here, connecting those 2 lobes. But these eosinophils have very large red or pinky colored granules. Then is the basophil, which is the final granular leukocyte. We have 3 granular leukocytes. And these granules are kind of variable in size. They don't really have a uniform size. They're all different sizes. So variable size granules, or variably sized granules. And these are basophilic. Basophilic means basic loving. So this means that they really stain very dark blue and purple with the basic dyes. They just really connect with that basic dye. So you can see it's very dark blue and purple, especially when you compare it to the eosinophils. The basophils, very, very dark granules, dark blue and purple. So very basic. Now these granules do commonly obscure the nucleus, which often has 2 lobes as well. Now, even though so-called agranular leukocytes possess cytoplasmic granules, these granules are just not visible under a light microscope because of their really small size and poor staining qualities. So these do still have granules, but we just can't really see them, so we pretend they're not there. So these granules are just very small, and they don't stain, or they don't hold stain. So these are agranular. So first off, we have the lymphocyte. Now the nucleus of the lymphocyte stains dark, and it's going to be round, and often it's slightly indented. So we can see the nucleus here. We can see this indent in the nucleus here as well. And the cytoplasm usually stains like a sky blue, and it just forms this small ring around the nucleus. So compared to the cell size overall, lymphocytes have huge nuclei. Covers almost the entire cell. But usually it's just a single lobe. Then we have monocytes. Now the nucleus of the monocyte is usually kidney-shaped. Or I guess some would say horseshoe-shaped. The cytoplasm is usually like a blue-gray and kind of looks a little foamy. You can kind of see here that the cytoplasm is kind of foamy looking, kind of. Now blood usually acts as a conduit for monocytes, which are going to migrate from the blood into the tissues. So monocytes are really cool. So they will actually leave the blood and go into tissues. In the tissues, though, this is where they will enlarge and differentiate into macrophages. So they kind of turn into macrophages. In a healthy body, some white blood cells, especially lymphocytes, can live for several months or even years, but most of them really only live for a few days. The main function is to combat invading microbes. That's their main function. Now, during a period of infection or invasion, though, phagocytic white blood cells may only live for a few hours. Red blood cells are contained within the bloodstream, but white blood cells, they can leave the bloodstream by a process that we call emigration. This is also called diapedesis. So here we have a diagram showing diapedesis. Remember, that's also known as emigration. So during diapedesis, the white blood cells will roll along the endothelium. They will stick to it, and then they squeeze between those endothelial cells. The precise signals that stimulate emigration through a particular blood vessel will vary for the different types of white blood cells. Here we have a neutrophil. Now, molecules that are known as adhesion molecules will help the white blood cells stick to the endothelium. Integrins on the neutrophil, these are a particular type of adhesion molecule. The term leukocytosis this is an increase in the number of white blood cells, and this is a normal protective response to stresses like invading microbes, strenuous exercise, even anesthesia and surgery. An abnormally low level of white blood cells, this is called leukopenia. This is almost never beneficial, and it may be caused by a couple things like shock or radiation. Even some chemotherapeutic agents can also cause leukopenia. A differential white blood cell count. This counts the 5 types of white blood cells, and this is to detect infection or inflammation, to determine the effects of possible poisoning by chemicals or drugs. It also will monitor blood disorders and the effects of chemotherapy, or also detect allergic reactions and parasitic infections. So this differential white blood cell count is quite important for detecting problems. Now, because each type of white blood cell plays a different role, determining the percentage of each type of white blood cell in the blood will assist in diagnosing the condition. So this table here lists the significance of both high and low white blood cell counts. So neutrophils, these are really important in bacterial infections and burns. So when we are looking at high counts of these types of white blood cells, this is what this might indicate. Lymphocytes have to do with viral infections. Monocytes are viral or fungal infections. Some leukemias, which are blood cancers, and some other chronic diseases as well. Eosinophils, their main role is allergic reactions, but they also have a little bit to do with parasitic infections as well, and some autoimmune diseases. And basophils also, main thing is allergic reactions. Besides the immature cell types that develop into erythrocytes and leukocytes, hemopoietic stem cells will also differentiate into cells that produce platelets. Now, under the influence of the hormone thrombopoietin, myeloid stem cells will develop into these megakaryocyte colony-forming cells that in turn develop into the precursor cells called megakaryoblast. Then those megakaryoblast transform into megakaryocytes, which are these really huge cells that splinter into about 2,000 to 3,000 different fragments. And each of these fragments is enclosed by a piece of the plasma membrane, and these are now called platelets. So each of those fragments is its own platelet. Now, platelets will break off from the megakaryocytes in red bone marrow, and then they enter into blood circulation. Platelets contain many vesicles, but they don't contain a nucleus. The vesicles that they contain granules, and these granules contain chemicals that, once released, they promote blood clotting. Sorry, the vesicles contain these chemicals. Now, platelets help stop blood loss from damaged blood vessels by forming a platelet plug. Now, platelets really have a short lifespan, usually only about 5 to 9 days. And then those aged and dead platelets are removed by fixed macrophages in the spleen and the liver. So these are macrophages that don't leave the spleen and liver. They aren't the type that travel in the bloodstream, but these are macrophages that stay in the spleen and liver. So then when those damaged and dead platelets travel through the spleen and liver in the bloodstream, those macrophages will dispose of them. Here is a table summarizing the red blood cells, erythrocytes, as well as the granular leukocytes. So here it gives you a really good idea of the number of each of these in the blood. This is not something that you have to memorize or anything. But it also does show you their characteristics and their functions. These are all things that we've talked about, but this is a nice table here that shows you kind of all together and in one place a nice summary here. So don't have to memorize the numbers in blood or anything, but the characteristics and functions are good to know. And then this part of the table shows the agranular leukocytes and the platelets. Again, characteristics and functions of each of these is good to remember. A bone marrow transplant, this is the replacement of cancerous or abnormal red bone marrow and replacing it with healthy bone marrow in order to establish normal blood cell counts. Now, in patients with cancer or certain genetic diseases, the defective red bone marrow is destroyed by high doses of chemotherapy and whole body radiation just before the transplant takes place. Usually this healthy red bone marrow from a healthy donor patient is taken from the iliac crest of the hip bone. Now, a more recent advance for obtaining stem cells involves a cord blood transplant. So stem cells would be obtained from the umbilical cord shortly after birth, and the stem cells are removed from the cord with a syringe, and then they are frozen for use in other patients. Okay, so let's talk about hemostasis. So hemostasis, not to be confused with the very similar term homeostasis. So this is hemostasis. This is a sequence of responses that stops bleeding. Now, when blood vessels are damaged or ruptured, the hemostatic response must be very quick and localized to the region of damage, and has to be also very carefully controlled in order to be effective. Now, there are three mechanisms that reduce blood loss. They are the vascular spasm, the platelet plug formation, and blood clotting, also known as coagulation. Now, when successful, hemostasis prevents hemorrhage. Hemorrhage is going to be the loss of a large amount of blood from the vessels. Now, hemostatic mechanisms can prevent hemorrhage from smaller blood vessels, but extensive hemorrhage from larger blood vessels usually requires medical intervention. Now, first, when arteries are damaged, the circularly arranged smooth muscle in their walls is going to contract immediately. Now, this is a reaction that's called vascular spasm. So this is where the circular smooth muscle in the blood vessel contracts really quickly. So this is going to reduce blood loss for several minutes to hours, and during this time, the other hemostatic mechanisms can really go into operation. So here we have platelet plug formation. So this is how a platelet plug forms. So here on the top picture here, this is when initially the platelets will contact and stick to parts of a damaged blood vessel, such as these collagen fibers of the connective tissue in the underlying damaged endothelial cells. So these little platelets here, just little purple things here that you could see in the blood vessel, they will actually kind of contact these pieces of collagen fibers that are sticking out of the damaged blood vessel, and they will stick to them. So this step is called platelet adhesion. Now, due to this adhesion, the platelets actually become activated. So this is where their characteristics really change quite dramatically. They extend lots of these projections that enable them to contact and interact with one another. You can see here that they kind of look like they have little projections, little like hands that are sticking out everywhere, kind of reaching out to other platelets, trying to make contact with other platelets and interact with each other. They also begin to liberate or release their contents of their vesicles. So this phase is called the platelet release reaction. So they are going to release ADP, serotonin, and thromboxane A2. So this liberated ADP and thromboxane A2, these play a major role by activating nearby platelets. So it's kind of like this chain reaction of activating all the platelets in the area. So we can kind of put this here, ADP and thromboxane. Activate the other nearby platelets. And then serotonin and thromboxane will both function as vasoconstrictors. So they cause this, they sustain the contraction of the vascular smooth muscle, which is going to work to decrease blood flow through that injured blood vessel. So this is going to be serotonin. So that all kind of happens in that second step, that platelet release reaction. Now, the release of ADP makes other platelets in the area very sticky. So ADP is also going to cause stickiness of nearby platelets. So this stickiness of the newly recruited and activated platelets causes them to adhere to the originally activated platelets. So we kind of get this gathering of platelets that's called platelet aggregation. And eventually the accumulation and attachment of large number of platelets forms this mass that we call a platelet plug. Now this plug kind of completely fills that blood vessel from one end to the other. So we're kind of getting this whole plugging thing happening here. Now this platelet plug is going to be very effective in preventing blood loss in a small blood vessel. So it can really stop blood loss completely in a small blood vessel if the hole in the blood vessel is not too large. Now, normally, blood remains in its liquid form for as long as it stays within blood vessels. If it's going to be taken from the body, though, like drawn with a needle and syringe, it's going to thicken and it forms a gel. Eventually, the gel separates from the liquid portion, and so we get this straw-colored liquid called serum. And serum is essentially going to be blood plasma minus the clotting proteins. So the gel part that separates from the liquid serum, this is going to be the blood clot. Now, blood clot consists of a network of insoluble protein fibers called fibrin. And fibrin kind of clots or traps the formed elements of blood. So we can see these fibrin threads here kind of trapping the red blood cell in this picture. Now, this process of gel formation called clotting or coagulation, this is a series of chemical reactions that begins in the formation of these fibrin threads. So you can kind of see it here in this top picture, kind of looking like a spider web almost of fibrin threads, kind of trapping all the formed elements, the red blood cells, platelets, white blood cells. Everything kind of gets trapped in this spider web of fibrin threads. Now, if blood clots too easily, the result can be thrombosis. Thrombosis is clotting in an undamaged blood vessel. On the other hand, if blood clotting takes too long, then hemorrhage can occur. Now, blood clotting involves several substances that are known as clotting factors, sometimes also called coagulation factors. And these are going to include calcium ions, some enzymes, and various molecules associated with platelets or released by damaged tissues. And these clotting factors are identified by Roman numerals that indicate their order of discovery. So it's kind of weird how they're numbered. They're numbered in the way that they were discovered or in the order that they were discovered, not necessarily by the order of their participation in the clotting process. So it's kind of seemingly random numbering of these clotting factors, but we'll see that in the next slide. Now, clotting is a really complex cascade of enzymatic reactions in which each clotting factor activates many molecules of the next one, but it happens in a very fixed sequence. Now, finally, a large quantity of the product, remember that's going to be the insoluble protein fibrin, is going to form. So this is kind of our end product of the clotting cascade is going to be that production of fibrin, that insoluble protein. So this is going to be a very, very important final step. Clotting can be divided into 3 different pathways. These are the extrinsic pathway, the intrinsic pathway, and the common pathway. So here we have the full blood clotting pathway or coagulation pathway. On the top left here we have the extrinsic pathway of blood clotting. This has fewer steps than the intrinsic pathway, and it occurs quite rapidly within a matter of seconds if the trauma is severe. Now it's named because a tissue protein called the tissue factor, or TF, which is also known as thromboplastin, it leaks into the blood from cells outside or extrinsic to blood vessels, and it initiates the formation of prothrombinase. So prothrombinase is down here at the bottom. Now, in the presence of calcium ions, tissue factor begins a sequence of reactions that ultimately activates clotting factor 10. Now, once factor 10 is activated, it combines with clotting factor 5 here in the presence of even more calcium ions to form the activated enzyme prothrombinase. So this completes the extrinsic pathway. Again here, this tissue factor is also known as thromboplastin. So that's going to be the whole extrinsic pathway. That's when there is extensive trauma, essentially breaking the entire blood vessel open so that tissue factor from the outside of the blood vessel, from the extrinsic, comes into and activates prothrombinase. Now with the intrinsic pathway here, this is a lot more complex than the extrinsic pathway, and it does occur more slowly, usually requiring several minutes. Now the intrinsic pathway is so named because its activators are within direct contact with blood or contained within the blood, or intrinsic to the blood vessel. Outside tissue damage is not needed for this pathway, whereas with the extrinsic pathway, outside tissue damage is needed. So in the intrinsic pathway, the contact with collagen fibers on the interior of the blood vessel is going to activate clotting factor 12. Now this begins a sequence of reactions that eventually activates clotting factor 10 down here. Platelets and platelet phospholipids and calcium will also participate in the activation. Sorry, not this bottom calcium, but these upper ones, these platelets, activated platelets, and platelet phospholipids in combination with calcium are going to work together with the activated clotting factor 12 to activate clotting factor 10. Now, once clotting factor 10 is activated, it combines with clotting factor 5 as well as calcium to activate the enzyme prothrombinase. This in the same way as it happens in the extrinsic pathway. So kind of down here, the two pathways are very similar. It's everything kind of above this line that is different between the extrinsic and intrinsic pathways. So the activation of prothrombinase is going to be the completion of both the extrinsic and intrinsic pathways. So again, with the intrinsic pathway, we have blood trauma that causes the endothelial cells to become damaged, exposing collagen fibers. And those exposed collagen fibers will activate clotting factor 12 as well as activate platelets, like we saw with the platelet plug formation. And those activated platelets will turn into platelet phospholipids or release phospholipids. And then the platelet phospholipids, calcium, and activated clotting factor 12 will all work together to activate clotting factor 10. Then clotting factor 10, 5, and extra calcium will all work together to activate the enzyme prothrombinase. Now, this formation of prothrombinase marks the beginning of the common pathway down here. Now, in this second stage of blood clotting, prothrombinase and calcium ions will catalyze the conversion of prothrombin into thrombin. So this is kind of our stage 2 of blood clotting. Prothrombin is also known as clotting factor 2. Now in the third stage, this is where we have thrombin in the presence of calcium converts fibrinogen, or clotting factor 1, which is soluble, and it converts it into these loose fibrin threads, which are insoluble. So the loose fibrin threads are insoluble, even though fibrinogen is soluble. So that thrombin in the presence of calcium is going to convert fibrinogen into loose fibrin threads. Thrombin also at the same time is going to activate clotting factor 13. Now once activated, that clotting factor 13 is going to strengthen and stabilize the fibrin threads into a really sturdy blood clot. So this is our final step in the coagulation step. So here we have a table of all of the clotting factors that are involved in coagulation. We have the Roman numeral number that they're known by, as well as the common name that they're called, their source. Most of them come from the liver. And then also really helpful, it shows the pathway that they're involved in. So some really important ones here that we do need to remember. Clotting factor 1, fibrinogen, an important one, as well as 2, prothrombin. Clotting factor 3, which is tissue factor, or thromboplastin. Remember, that's going to be the first clotting factor involved in the extrinsic pathway. That's an important one. Calcium ions, these are kind of given their own number here too, which is number 4. Calcium ions are involved throughout the entirety of coagulation. Clotting factor 5, also very important. And then clotting factor 10, 12, and 13 would be our other major clotting factors that are very important to us that we need to remember, especially if you're going to be explaining the whole coagulation pathway in an exam. Those are definitely the 8 or so, I guess, clotting factors that you should know. clot retraction. This is going to be the consolidation or tightening of the fibrin clot. Now, the fibrin threads attached to the damaged surfaces of the blood vessel will gradually contract as the platelets pull on them. Normal blood clotting really depends on adequate levels of vitamin K in the body. Even though vitamin K isn't directly involved in actual clot formation, it is required for the synthesis of 4 major clotting factors. So vitamin K, very, very important. Now, blood clotting involves the amplification and positive feedback cycles. A clot has a tendency to enlarge, creating the potential for impairment of blood flow through undamaged blood vessels. And because of this, we have this really important system in our body called the fibrinolytic system. And the fibrinolytic system is going to dissolve small inappropriate clots. It's also going to dissolve clots at a site of damage once that damage is repaired. Now, this dissolving of clot, removal of clots once damage is repaired, is going to be completed by the activated enzyme plasmin. So very important little pathway, very important little enzyme that we need to know about that really helps to get rid of those clots once we no longer need them, or get rid of clots in blood vessels that may come up even when they're not needed. So let's talk about blood groups a little bit. So the surfaces of erythrocytes contain a genetically determined assortment of antigens that are composed of glycoproteins and glycolipids. So these glycoproteins and glycolipids are called antigens on the erythrocytes. These are also called agglutinogens. Now these occur in very characteristic combinations. Now, based on the presence or absence of various antigens, blood is characterized into different blood groups. Within a blood group, there may be 2 or more blood types. Now, there are at least 24 blood groups and more than 100 antigens that can be detected on the surface of red blood cells. So for this class, we're just going to discuss 2 major blood groups. These are ABO blood groups and RH blood groups. So first off, the ABO blood group. This is based on 2 glycolipid antigens called A or B. Now, people who have red blood cells that display only antigen A will have type A blood. So if they display antigen A on their red blood cells, they'll have type A blood. Now, those who only have B antigens on their red blood cells will have type B blood. Individuals who have both A and B antigens on their red blood cells will be type AB. And those that have neither A nor B antigens on their red blood cells will be a type O. Now, blood plasma contains antibodies usually. And these are usually called agglutinins. Sorry, agglutinins here. We usually just call them antibodies though. And these will react with the A or B antigens if they are mixed. So anti-A antibodies, remember these are in the plasma, will react with the antigen A on red blood cells. And anti-B antibodies in the plasma will react with antigen B on red blood cells. This is a negative reaction. Now the antibodies present in each of the 4 blood types are shown here. So this is within the plasma. This looks like the anti-B antibody. This is the anti-A antibody. And one very important thing is that you do not have the antibodies that react with the antigens of your own red blood cells. But you do have antibodies for antigens that your red blood cells lack. So what that means is that if you have type B blood, for example, your red blood cells will have B antigens all over them. Now because of this, you're not going to have any of these anti-B antibodies in your blood, because if you did, these antibodies would attack all your red blood cells and would kill them. On the other hand though, if you do have type B blood, your red blood cells are going to show or display that B antigen, and you will have anti-A antibodies within your blood plasma. If you have type A blood, your red blood cells will have A antigens all over them, and you're going to have anti-B antibodies in your blood plasma. Now these antibodies will not attack your own red blood cells, but if you do have other blood cells that come into your blood that have, say, your type A, and you have blood cells that come into your blood that have B antigens on them, the anti-B antibodies within your blood will attack those foreign blood cells that have B antigens on them. If your red blood cells have both A and B antigens on their surface, you're going to be type AB, and you won't have anti-B or anti-A antibodies in your blood plasma, because if you did, they would attack your red blood cells. If your red blood cells don't show either A nor B antigens, you are type O. And in that case, you're going to have both anti-A and anti-B antibodies within your blood plasma. Now, despite the differences in red blood cell antigens reflected in these blood group systems, blood is actually the most easily shared of all the human tissues. So people with AB blood type, they don't have any anti-A or anti-B antibodies in their blood plasma. So their antibodies, they don't have any anti-A or anti-B antibodies in their blood plasma. Now, because of this, AB blood types are called universal recipients. And this is because theoretically they can receive blood from all 4 blood types. They have no antibodies to attack antigens on donated red blood cells. Type O blood type, they have neither A or B antigens on their red blood cells, and because of this, they're sometimes called the universal donor. And this is because they theoretically can donate blood to all 4 blood types. Type O persons requiring blood, though, they can only accept blood from other type O blood groups. And this is because they have both anti-A and anti-B antibodies in their blood plasma. So if they get blood from an A blood type, their blood plasma is going to attack and kill those donated red blood cells. Now, to avoid blood type mismatches, lab technicians will type the patient's blood and then either cross-match it to potential donor blood or screen it for the presence of antibodies. In this procedure for ABO blood typing, single drops of blood are mixed with different antiserums. And antiserums are going to be solutions that contain antibodies. So they're either going to contain anti-A antibodies or anti-B antibodies. So here we have the blue anti-A serum and yellow anti-B serum. So again, anti-A serum will be a solution containing anti-A antibodies. And the anti-B serum will be a solution containing anti-B antibodies. So here we just have this column of untreated blood. In this column, we have blood that is treated with the anti-A serum. Now, one drop of blood is mixed with the anti-A serum, which contains those anti-A antibodies that will agglutinate with the red blood cells that possess anti-A antigens. This term was on the previous slide, but agglutinate means to, this is when the antibodies will attach to the red blood cells and clump, making them completely ineffective, pretty much removing those red blood cells from being functional. So when we mix a drop of blood with the anti-A serum, if there are red blood cells that have A antigens on them, the anti-A antibodies will react with them and clump together with the red blood cells. So that's what we see with this reaction and this reaction. They're all clumped together. You can see these like clumps of red blood cells. They are completely non-functional now. These other two, though, these had no reaction. So we would say that this top one, because it was treated with the anti-A serum, this top one has red blood cells in it that have A antigens, as well as this one. So we know that the blood contains A antigens. Now another drop of that same blood is mixed with anti-B serum. Now remember, these contain anti-B antibodies, and they're going to agglutinate with red blood cells that possess anti-B antigen, or they possess B antigens, sorry. So the anti-B serum contains anti-B antibodies that will agglutinate with red blood cells that have B antigens on them. So you can see that agglutination happening here and here. So when we take all of this into account, all this information, the top row agglutinated with anti-A serum but did not agglutinate with anti-B serum. So the blood type would be type A, because we know that those red blood cells have A antigens on them but no B antigens. Our second row here, this blood did not agglutinate with the anti-A serum, but it did agglutinate with the anti-B serum. So we would say that blood would be type B blood. Those red blood cells have B antigens on them. Our third row, this blood agglutinated both with the anti-A and anti-B serum. So these red blood cells contain both A antigens and B antigens. So this would be an AB blood type. And then in our last row here, we had no agglutination with anti-A, no agglutination with anti-B serum. And so we could deduce that these red blood cells have neither A antigens nor B antigens on their surface. So they would be a type O blood type. Now pretty much the same procedure, or a very similar procedure, is done for determining Rh factor. So this is where a drop of blood is mixed with antiserum containing antibodies that will agglutinate with red blood cells displaying Rh antigens. Now if the blood agglutinates, then it is Rh positive, and if no agglutination occurs, it is Rh negative. So this is where you get like your positive or negative that comes after your blood type letter. So like in A positive blood type is going to have A antigens and also be positive for Rh antigens. AB negative, for example, is going to have both A and B antigens, but no Rh antigens on the red blood cells. The most common problem with Rh incompatibility may arise during pregnancy. Now, normally there's no direct contact that occurs between the maternal and fetal blood while a woman is pregnant. However, a small amount of Rh positive blood can leak from the fetus through the placenta into the bloodstream of an Rh negative mother. So this only occurs if the baby's Rh positive and the mother is Rh negative. In this case, if a small amount of that Rh positive blood leaks into the maternal blood through the placenta, then the mother will start to make anti-Rh antibodies. Now, because the greatest possibility of fetal blood leakage into maternal circulation occurs during delivery, the first baby is usually not affected. If the mother becomes pregnant again, however, her anti-Rh antibodies can cross the placenta and enter the bloodstream of the second fetus. Now, if that fetus is Rh negative, there's no problem because the Rh negative blood doesn't have the Rh antigen for those antibodies to attack. If the second fetus is Rh positive, however, if she has a second pregnancy with an Rh positive baby, then agglutination and hemolysis will occur. This is brought on by that fetal maternal incompatibility. Hemolysis is the exploding or destruction of red blood cells. So this is what happens in that case. During that first pregnancy, we have an Rh positive fetus with an Rh negative mother, and some of those antigens from the fetus will cross into the mother through the placenta. And this will cause the mother's immune system to create those anti-Rh antibodies. This is between the pregnancies. Then, if there is another Rh positive fetus in a second pregnancy, for example, those antibodies will go and attack the fetus. Essentially, it sees it as a foreign, kind of like an infection. Now, in these cases, there are things that can be done. So an injection of anti-Rh antibodies called anti-Rh gamma globulin can be given to prevent this. So an Rh negative woman should receive this injection before delivery and also soon after delivery. And these antibodies will bind to and inactivate the fetal Rh antigens before the mother's immune system can respond to the foreign antigens by producing her own anti-Rh antibodies. So this would occur before and after the first pregnancy. The mother would get injections of the anti-Rh gamma globulin. So we're going to briefly talk about a disorder of the blood, homeostatic imbalance. So first off, anemia is a condition in where the oxygen carrying capacity of the blood is reduced. Sickle cell disease, this is a genetic anemia. Now, the red blood cells of a person with sickle cell disease contain hemoglobin S, abbreviated HbS. Now, when HbS gives up oxygen to the interstitial fluid, it forms into these really long, stiff, rod-like structures that will actually bend the erythrocyte into this sickle shape. So here we can see these really strangely shaped red blood cells, especially compared to a normal shaped red blood cell. And these sickle cells will actually rupture very, very easily, and they die in just about 10 to 20 days. So this reduces the oxygen carrying capacity of the person with sickle cell disease quite significantly, because we have these abnormal cells, this disorder that causes these cells to die very quickly. And that's going to be the end of chapter 17.